Thank you, Esteban. Now, if at any time you can't hear me or something goes on, if you or Elsie will just raise your hand and wave at me, then I'll try to figure out what's going on. I just wanted to correct one thing in the introduction because it's important to how I'm going to tell you this story today. And that is, I'm not a professor at the University of Alabama, not a tenured professor. Formally, I'm retired.
Sorry.
No, it's not. It's actually part of the story. I am a research professor that allows me to continue to do some work there. But the story is that I've had to make that transition between academics and industry. It's not an easy transition, and I'm trying to exist in both worlds. And I'm going to show you why today. Before I do that, I want to thank you for the invitation to speak. I'm very disappointed I can't actually be in Chile for the meeting. I remember going to Joao's in Porto, and traveling and meeting people is really a big part of these meetings. But on the other hand, I'm sure that I'm speaking to a lot more people now than I would be able to travel. So I'm going to start and talk maybe less about ionic liquids at the beginning and talk a little bit. Let's make sure I can do this. There we go. Let's start with why I'm working with ionic liquids. I want to remind people, and now in the United States, I'm allowed to say we're all about global sustainability, but some of us have been working in the field of green chemistry for quite some time, where we're trying to make the world better by the design and development of better chemical processes. This field started before ionic liquids, but had an interesting intersection. My first paper, those of you on editorial boards will know when you have to put up an ad, but my first paper on ionic liquids was in ChemCom in 1998, and it had to do with separations, an ionic liquid that was immiscible with water. And then 2 years later, we had a workshop in Crete. Notice 2000. This is the late Professor Ken Seddon. Actually, a lot of you could probably recognize yourselves in this picture, but it was called Green Industrial Applications of Ionic Liquids. And there were two outcomes, an academic and a motivational one. The academic outcome was, man, this is interesting. We really need to study everything there is about ionic liquids. But the other one was, combined with green chemistry, this is an opportunity to work with engineers and business. That last one is the one that I've been looking at for quite some time, and it will be the subject of today's story. Here's an example of green chemistry. A raw material or feedstock should be renewable. Okay, everybody knows this one now. However, the question I want to ask is the goal to make nature-derived chemicals so we can make more plastics, or is it to use natural polymers to replace plastics? The first one keeps the chemical enterprise going, but also generates a lot of waste that the chemical enterprise is going to generate. And the new plastics are not necessarily any more biodegradable. So I want to look a little bit today at that second one, because most of you realize the world is drowning in plastics. We hear a lot about CO2 and global warming, but you should look at the amount of plastic that's going in the ocean. It wasn't always this way. So I'm going to tell you a brief little story about plastics for those of you who don't think that biopolymers can replace them. The first plastic product was Bakelite in 1907, and it was a very high-end, very expensive kitchenware or for jewelry at the time, even though the polymerization reaction was discovered, I think, in Germany in the 1880s, I believe. It wasn't until 1907 that there was a commercial product. But now, by 2050, there's going to be more plastic in the ocean than fish. and it's a huge, huge market. It wasn't always the case. Around the 20s, late 19s and early 1920s, Henry Ford was trying to use plants for the polymeric materials in cars. He even worked on the hemp car. You know, hemp is back in the United States, by the way. We can grow it again. This was instead of plastics, but today we have 400, 500, 600 different parts of plastics in virtually every car. And you start having to ask yourself, why did we not keep using the plants? I like this one. George Washington Carver was a professor in Alabama at Tuskegee Institute, and he actually worked with Henry Ford on the soybean car. Again, polymers and materials from plants to make our automobiles. You got to think about that. So what happened? Well, the biggest thing that happened was war. The US lost its source of rubber, and so polymer chemistry became a really big thing. There were huge investments from the government. They got really economy of scale, and because they were cheap, there was a lot of money to work on them. There was innovation, and we had product after product after product that would come out of them. So now we have to find a way to be able to take biomass, I think, and instead of turning it into chemicals, turn it directly into products with as little chemical derivatization as possible. It's also going to take huge investments, huge economy of scale, and lots of innovation to get there. I am very, very hopeful that it's not going to take a war. Here's the challenge that we face with the most common biomass, trees and grasses, lignocellulosic biomass. It's a separations challenge. Cellulose is often the polymer people want. Hemicellulose is also useful in there. And then lignin, that still has the reputation of not being worth anything, but you can do a lot with it. The current industrial process is kraft pulping. And I'm not going to go through the whole thing, but to break it down, it essentially destroys the lignin and the hemicelluloses and tries to leave the cellulose alone. So it's a destructive separation. Now you finally get to see some ionic liquids, an old-fashioned ionic liquid slide. I'm not going to talk to you today about what ionic liquids are. I'm really barely going to talk about ionic liquids at all, at least to this audience, but how you can use them. I heard the pre-talk discussion on separations. Well, that's exactly where I got started, using these for separations. But there was an interesting separation that we found when cellulose, pure cellulose, would make a solution dissolve in an ionic liquid. We used butyl methylimidazolium chloride in the beginning. But to us, that was amazing, because if you can dissolve it and reconstitute it really simply, there are all sorts of different things you can make from it. And notice I'm not talking about making money yet. I am just talking about the technological feasibility of making materials directly from cellulose. And then we decided to look at the pulping process and found that to some extent we could even do trees or other grasses. I know this is an old story now, but think about this with, in this case, ethyl methylimidazolium acetate with or without a catalyst. You can separate cellulose, hemicellulose, and different fractions of lignin. I think that's important. I don't know of any other solvent system that can do that. Kraft doesn't do this. Kraft modifies the lignin and breaks it up. So nonetheless, a lot of those people talking about using cellulose today are talking about taking it from the Kraft process. Well, it's cheap. It's known. It's been there for 150 years. And we're doing green chemistry with the cellulose, so we don't have to worry about how dirty and all the waste that comes out of this. That may work today, but it's not always going to work, because if you could just take any biomass and relatively easily dissolve it, even if the solvent is expensive, you can then get high molecular weight polymers. You can get extractives, and my goodness, I'm sure a lot of people on the call have seen or are doing all the extractions of natural products and everything you can do with ionic liquids and biomass these days. So if you don't pulp it, then the cellulose, the lignin, the hemicellulose are higher molecular weight polymers. It seems to me as a chemist that should be worth something. So now we're back to this, okay, what can we make instead of chemicals from the biopolymers? You don't have to use covalent organic chemistry to make things. You need to be able to dissolve them and reform them. You can make fibers. A lot of great work coming out of Finland in a whole process for making cellulosic fibers from ionic liquids. You can make beads for specialty purposes like medicines. You can make membranes. Go look up how they make cellophane today, and then think about taking a tree and dissolving it and making a film directly from that. Those are the technologies that I'm talking about trying to be able to do. Almost any biopolymer in almost any shape with almost any additive. Now notice I haven't had to say one thing about making money from this. The technology to do it is there. Is there enough motivation? Well, let me talk about commercially feasible then, because that's the next one. And I told you why synthetic polymers were commercially feasible. They've got precise molecular weights and molecular weight ranges that they can make, and they're very low cost, pennies on the kilogram, because they now have huge economies of scale. So if we're going to enter into the polymer market, we have to have controlled molecular weight. We have to have consistency, which you often don't get from biomass. And you have to have a large supply. Now, there are celluloses out there where you can get a specific molecular weight, but the chemical companies that are doing that essentially are degrading the polymers, they're derivatizing them and fractionating them. So we were curious if we could control the molecular weight by the age of the organism. Now, this is a little bit tough to think about for a tree, because for a tree, once you cut it down, it doesn't continue to grow. But if you were thinking about this as a strategy, you might grow different types of trees and then take the ones at different ages. It's going to be hard, but maybe there's some other ways. If you look at the evidence, eucalyptus trees, some of these, there is some evidence that the molecular weight will increase over time. Nobody really studies it that much, probably because you have to kill the tree mostly in order to get the data. However, now we come to the title slide. Wouldn't it be easier to control biopolymers obtained from animals by age? We actually grow a lot of our food based on the age of the animal. Also, animals like shrimp have a different polymer, in this case, chitin, and chitin happens to be very valuable. Before I tell you how valuable, I want you to notice that, the difference between chitin and cellulose is really this amine group. What's useful in medicine for chitin today is the deacetylated version, which is the amino version, where you can have a polyvalent polymer. And that value of chitin has translated into big business. So many of you have seen this before, but we got interested in chitin in 2010 in Bayou La Batterie. This is Forrest Gump. not a real resident of Alabama, but pretty famous. And we found all of the shrimp shell. I want you to think about that, shrimp shell. Because during its life, but we only tend to worry about the last one that comes off the animal. All right, now for commercial viability, huge economic driver. Here's a polymer that has a $63 billion market, and it's used in drug delivery, agrochemicals, but it can also be used in low-end applications as well. And as many of you know, it's also a food additive. And I always found it interesting that there are still today, I've been talking about this for a long time, there are no chitin producers in North America. It's all imported. And you know, with the new president, that may not cut it anymore. But let me tell you why, for one reason. The process for getting chitin today is like the process for getting cellulose from trees. It's pulped. In other words, you use acid and caustic to get rid of the minerals, to get rid of the proteins, anything else, and just leave the chitin alone. So the chitin is gotten by destroying everything around it. And then if you want to use it in medicine, you have to further purify it. But those steps cause degradation in that polymer. Excuse me. Again, now the what if you could take the crustacean shell in a one-step process exactly the same as using trees and get a high purity chitin with a high molecular weight, much higher than this. Would that be worth something? So now, instead of having just high molecular weight polymers like cellulose and lignin, you have high molecular weight and valuable polymers that you might be able to make commercially successful. And of course, just like cellulose, you can make fibers, although now these fibers are naturally antibacterial, can be used in medicine such as sutures. You can make films as I showed you with cellulose, but now again you have films that can be embedded in the body and decompose, releasing medicines. You can make cosmetic beads instead of plastics to carry cosmetics or cosmeceutical type materials. And you can make nanofiber masks. I would like to tell you that I've been able to take this one further than I have, but what we are actually working on is a nanofibrous mesh that can act as a face mask. And we all know why face masks are important today, but the fact that we can electrospin this into a high surface area material that can be used in the filter compartment of many of today's face masks, I think is very important. And I hope that we can make some progress on that particular one because it's important. And so now you have essentially waste that we get from shrimp and we make materials, but there's still more to look at here. What if we could control the molecular weight and get economy of scale, larger supply, by controlling the age of the shrimp? Many of you may not have seen this paper yet. It just came out in Green Chemistry at the end of last year. But I wanted to just briefly show you a strategy that the Ionic Liquids allowed us to do to see if we could maximize the chitin obtained from a given shrimp and control the molecular weight. Well, the first part of this story is we had to look at shrimp that were grown in ponds rather than live caught because we wanted to catch their molts. Shrimp, as they grow, throw off their shell. It's called a molt. And then they grow another one on top of it. And we wanted to know if we could capture all those. So we collaborated with Global Blue Technology in Rockport, Texas, and they had a zero waste, zero discharge shrimp farm. They used a specific hybrid shrimp that they had grown. And in this one facility, they could grow 10 million pounds of shrimp per year. Now, right now, they harvest the shrimp based upon the market for the meat. So certain restaurants in Washington wanted huge shrimp, so they would grow them like 50 grams. Other markets wanted smaller shrimp, so they would grow them maybe 10 to 20 grams and harvest them. But we were curious about what would happen if you harvested their shells instead. And so they organized raceways for us in the ponds, and as the molts settled to the bottom, they collected them and they were sorted by body weight. And we were able to get 3 different weights: the young juveniles, the middle juveniles, and then the adults. We didn't get quite as much of this last one, so didn't have a complete study. But for comparison, we also got the peel that was taken off the fully grown animal. And the first thing we did is to use a traditional blackened sorts method to see how much chitin was present. In the molts, there was not as much chitin as the peel, but that's a matter of biology. The animal keeps some of the chitin on its soft shell so that it can grow a hard shell around it. But nonetheless, there's a lot of chitin in those shells, and it does appear to increase with the age, which may have to do more with the fact that it's bigger. So we looked at extracting the chitin using ethylmethylamidazolium acetate, the same technique I had mentioned before, and we found that it wasn't not easier, but it wasn't harder to extract. About 70%, 60 to 70% of the chitin was extracted in one step from this. But what makes this fascinating is that you're looking at now all of the added mass, all of the added biomass that normally would just degrade. We've captured it. So we started looking at how much, if you captured everything, how much you could get. So we made a very conservative estimate, and we estimated that at least 8 times as much chitin by mass could be extracted from a single shrimp by extracting all of its molts and the final peel, as opposed to just the peel. Now, that's something to think about supply. They had 10 million pounds of shrimp. I know a lot of that counts the animal, but think about how much mass of chitin that we would be able to collect. So we're starting now to think, okay, we can handle the supply. We can handle the chitin supply. But now let's take a look at molecular weight. There was a separate green chemistry paper that published a month or two before this one, and Dr. Julia Shamshina had developed a method to determine the molecular weight of chitin dissolved in the ionic liquid, which was a big step for us. And I'm not going to go through all the details, but the bottom line is, yes, you can control the molecular weight by the age of the shrimp. So the youngest shrimp, we got an average of around 3.9 megadaltons for the average molecular weight. The middle juveniles, it was up to 22.6. That's just amazing. With live caught, which is an average, of course, of every shrimp that comes off the boat, you don't know what it is. The live caught, we got 3.9 megadaltons, and practical grade, which is what we buy commercially, which has been pulped, we got 1.6. So high molecular weight polymers. We felt anyway, this was proof for what we've been saying for a long time from indirect evidence. The molecular weight of the polymers that are dissolved in the ionic liquid rather than pulped in a chemical process are much higher molecular weight. So what does that mean? That means that we probably can solve the supply problem, because the chitin content, even though it's less than in the peel, if you collect all the molts, and there might be 50 in a given shrimp's life, you're going to get at least 8 times more chitin by using it all. And if you want a molecular weight of a particular value, high to low to smaller, then you can get it by harvesting the molts only in certain age ranges, and the shrimp continues to grow. And you can get the other molecular weights as it goes through other ages. So I actually think that the shrimp and the chitinous biomass is a much more valuable biomass reserve because you can control it. Now, this means we can't harvest it from the sea because you can't control the ages and you can't get the molts. But if we do this in like these zero discharge ponds. All right, so what I'm trying to tell you is technical feasibility has never been a problem. Ionic liquids are great at allowing you to do things that nothing else can do. Commercial feasibility is still a problem. We still have to get to market. And I'm going to show you the bumps along the way for what we are currently have done and what we're currently doing. One way to get to market would be just to go after high value products, medical sutures, drug delivery devices, face masks, cosmetics, and that's still pretty attractive to me to look at all of the different products you can take to market. But if you take them to market, you still have to have a supply, and it has to be a consistent supply. So you do also have to look at how are you going to scale it up? And for a while, I tried to do both, and I guess to some extent we still are. At the University of Alabama, before I went to McGill, we took our 3-liter reactor and we developed, with Department of Energy funding, a 20-liter reactor. And we also developed the electrospinning that allows us to do the nanofiltration mask. But you still, to get to commercial scale, you have to go big. And even 500 liters, or the commercial scale, that's a lot of money because it is a chemical process. Well, it's really engineering at this point. So we developed the 20-liter reactor, and during this time, we thought we were doing pretty good, but we still have to go to the next phase, raise money to really go big. So I'm going to come back to the story of Global Blue Technology. You did hear me mention that Global Blue Technology was in Rockport, Texas, I believe. Well, Rockport, Texas was also the landing point for Hurricane Harvey. So this facility was completely wiped out, which interrupted our shrimp molting study a bit, but we had enough to be able to finish it. Before it was wiped out, though, we formed a joint venture with them to form a company that was called Mary Signum Limited. And they raised the money to actually scale it up. So these are 1, 2, 3, 4 1,200-liter reactors. This is one of the 200 kilowatt microwaves. And they demonstrated that worked. See, the Ionic liquid was in here. This is the crash tank for those of you that know about the coagulation step. There was biomass processing in here. There's centrifuge and filtration in here. It was an interesting facility. I even worked with Roland Kalb at ProIonics, and they were scaling up the Ionic liquid by MAD. And this is 5 tons of Ionic liquid, ethylmethylamidazolium acetate, provided by ProIonics at a very high cost. Now, I hope Roland's on the webinar because he sent out a flyer, I think this week. Many of you may have gotten it, but I really liked this one. They're going to essentially do the Ionic liquid recovery step. This is very important. We've been talking about this since NATO meeting in Crete, and certainly since the biomass materials have gone. a lot of the commercial viability is not just the value of the biomass, but it's how efficiently you can extract and recover. And I really thought it was just a matter of time before one of the top ionic liquid companies actually started providing this as a service. And I will tell you this, excuse me, this really is an important part of the entire process. Now back to Mary Signum, they won 2018 Green Chemistry Challenge Award. I even got a plaque with 525 solutions for that. So I thought things were going pretty well. However, this is one of those stories about green chemistry meets world, or green chemistry meets reality. Global Blue Technology was controlled by a man named David Wills. So first there was Hurricane Harvey, which wiped out Global Blue Technologies. But David Wills was also, because they raised all the money, the primary shareholder in Mary Signum Limited. Now, Mary Signum Limited in 2019 was essentially bought out by a publicly traded company. In fact, they still own it, the Ross Group. This is a UK, whatever their highest board is on their stock exchange. But David Wills was convicted of a rather bad offense, and during 2019 was sentenced to life in prison. And he was supposed to be running Mary Signum Limited for the Ross Group as part of an agreement. So essentially, Mary Signum Limited is just a name. Now they're closing it down. We've formed a joint venture with RGP to try and pick this up in a much, much smaller way, starting from the extraction. In fact, we're back to the startup way. And the startup way is a problem for most startups. I know, Joao, I don't know if Mara's on the webinar, but when you start these startups, there are barriers. If the university is not going to give you free lab space, you need chemical laboratories, you need prototyping space, you need equipment, you need trained personnel. Shared facilities would be nice because that would lower the cost for all the various startups. So what I've done in work is to try to work with some of our startups in a consortium for green manufacturing to find ways to work together. Now, I've started with companies that I'm involved with, but that doesn't mean that it can't grow. You know about 525 Solutions and Dr. Garreau. You may not know about Ionic, which is a company in Laramie, Wyoming, that I founded with my daughter, Kristen, my son-in-law, Caleb, my sister-in-law, Gabby. So it is a family company to look at ionic liquids, and we've gotten grant. We just got a grant from the state of Wyoming to develop the chitin into the face mask material. But they're in the same situation we are. Startups don't have space and they don't have money. I also started a company with a former postdoc, Dr. Paula Burton, and her company is in Calgary, Canada. Calagua is looking at cleaning up oil field waste using ionic liquids. So in Tuscaloosa, to try to overcome this, I bought a couple of buildings with enough warehouse, that's almost warehouse type space. And we're renovating these for the lab space that could hold maybe up to 8 companies. It is expensive, so the renovations are going very slow, but we can actually do the work in these buildings. And we do still try to work with the University of Alabama, but most of you can understand what happens if a university out of your control decides they're going to close every lab for months or more, as many of you have experienced with the pandemic. So I felt it was much better to be able to control the space and control the expenses. So we formed a JV with the Ross Group. We've moved a lot of the research equipment assets of Mary Signum down to Tuscaloosa. And we're working across this startup platform to try to build synergies for the development of new technologies. And you probably noticed all of those companies. Can y'all still hear me?
Yeah.
Yeah, sorry.
Okay, where did I go mute?
I don't know. It went mute,
Oh,
but it's okay
okay.
now.
I just had a message saying that I was muted. Okay. Well, anyway, I'm almost done. We'll get to the interesting questions part. The idea is that we have to work really hard, and I believe all of you know what that means. Academics have to work really hard all the time, small businesses as well, and it's always the same problem. We're going to have to keep raising the money to do it. I'm actually excited about doing it because it really is this idea of scientific discovery where academics love to play, developing the intellectual property, and then commercialization. So we have to invent. That can be done easily at universities. Develop and prototype, not so easy at universities. And then the commercialization, you really need partners that have to have money. And then we can transfer those new sustainable technologies. Some people ask me why I didn't just stay a tenured professor. It's because making a transition, trying to transition things out of a university often is much more difficult if you stay 100% within the university. So I've tried to straddle both, and that means in some cases you can't have the perks like tenure or salary or things like that from a university to be able to try to make that transition. But I'm always looking for partners who would like to join us in this effort, and investors too one day. Thank you very much for your attention.
Well, thank you very much, Professor Robin Rogers, for that nice presentation. Now we have space for questions. Please raise the blue hand. Then I can give you the stage for question. Boyan Iliev can unmute yourself and ask the question.
Oh, the host may have to allow everybody to unmute.
Yeah, let me see. Boyan.
Yay! Wonderful. Thank you so much. Robin, excellent presentation as always. Just a small note. I mean, it's wonderful, of course, that the Ionic Liquid can be recycled. That's basically the only way you can actually build a sustainable and economically viable process. And just a small note, we received exactly for that, let's say, concept, rent an Ionic Liquid, we called it at the time, the Innovation Prize Profiles in Sustainable Chemistry Award at the Infomex 2010, a while ago with Tom, while he was still actually at your premises. And we've been doing it for the past 3 to 4 years, actually, I must admit, only really and effectively for 2 or 3, I must admit, also smaller processes than, of course, this one. But this is, again, really the way in order to achieve a really sustainable and commercially viable process. Thank you.
I think you're right. That concept really has been knocking around for a while, to rent or allow the companies to buy back. Never really been commercially viable. What I liked about what Roland and Ionic was doing is that I think that they were actually bringing in their engineering partners and trying to help companies to put a full package with pro forma and costing and everything up before they actually had to build a plant. And having worked with Mary Signum, that's a big one. Like, you know, we can ask you for cost of ionic liquids or proionics for cost of ionic liquids or even cost of recycling. But if you can engineer the recycling into the process, and the processes are all going to be different, it's going to require more than just the cost. It's going to require, you know, it's like building an oil well. You're going to have to go in and build it for the site. And that, I think, was missing. Now, I don't think Proionics has done it yet. Maybe they have. Maybe y'all will do it as well, but it would be welcome.
Yeah, I mean, this is generally what we're trying to do, depending, of course, on the size and the customers, but that's the general idea.
Well, that's the fun part about having worked with Mary Signum is I got to see it. I thought they were making a lot of bad decisions. I was not involved, so I can say that. Or I could say I was involved and say it was bad too, I suppose. But they were making bad decisions that I feel were based on not really understanding what an ionic liquid was and just thinking of it as a chemical. So the guy who was overseeing and trying to run this was a former oil field guy and thought that all you had to do is just build it big and that's it.
Yeah, that would be nice if it worked like that. Yeah, we see that often as well. Thanks so much.
You're welcome. I'm going to answer this one question that I was asked if I have any samples of the chitin molecular weight available right now. No, you can send me an email and I can go back and look. But the main problem is that the shrimp farm blew away. I mean, it was destroyed, and we only had what we had to do the paper. So we know it's possible, but we don't know any shrimp farm that's actually collecting the molds. In fact, most of you know shrimp farming, which is big in Alabama, is very environmentally damaging because they don't collect the waste and they allow it to overflow. Well, they don't have any way to collect the molds. So you would literally need that pond, but there is a company, I don't think David Wills from prison is allowed to run it, but there is still a company with that facility that they say they're going to rebuild it, but I don't know how long that will be.
Okay, thank you very much.
One quick question.
Yeah.
I just remembered something. So you can do this having two molecular weights by buying chitin from shrimp chitin from overseas, or here it's overseas, and by extracting your own from live caught shrimp. You would at least have two different molecular weights to look at.
Super. Professor João Coutinho.
Good morning.
Hi, Robin. Thank you, Stephen, for organizing this seminar. I hope that there will be many more. I do think that the Ionic Liquid community is missing a meeting point, and I hope that this will be, if not the meeting point, at least one of them. Robin, wonderful to see you again after all these months. Your story, your story is quite inspiring for all of us, but it's also very depressing. I won't explain why it is inspiring, but the depressing side makes you shy of doing or attempting to do what you have been trying to do these last few years. So having faced all these troubles and knowing that not everybody has the same tenacity, the will to continue fighting after all the unsuccesses. From what you have learned, how far would you advise that we should go?
I think you have to take a personal motivation. I think you're correct that I do this because I want to do this. This is something that I've always enjoyed and I'm willing to push it through. But I don't think what I do is any different than being a professor. You always have to fight for funding, you have to fight for the right teaching loads, you have to fight for students, and a lot of times the university will do and say things that make you not very encouraged to want to do those things, but you still do it. When I look at what Mara is doing, for example, I think it's different. What I'm trying to do is to change the chemical industry. What Mara could do is to make a fortune and have an incredibly important product. So that's what most startup companies would do. Not follow my story. They would pick a product and run with it. And I think within the field of ionic liquids, many people can do that. My story is slower because it's a different long-term goal. It's like, look at the last 4 years in the United States. I mean, that would have turned off anybody from doing anything, but still people are trying, and it's the important thing to do. Besides, if we work out some of these little things like ionic liquid recycle and cost, I tried to emphasize this many times. Ionic liquids allow new technologies. Plastics used to be the new technology. So think about what would happen if all of a sudden there was incredible need and only ionic liquids could provide the answer. Cost wouldn't be the issue. It's like, and I hate to use a bad example, but the Manhattan Project and the nuclear bombs and everything else only happened because a huge amount of money and what was perceived as a need went into it. Plastics, again, based upon war, the innovation came because of huge investments. So is President Biden going to make the same sort of Manhattan-style investment in climate change? If he does, there will be funding for startup companies to do the types of things that I want to do. There's been no appetite for green chemistry in the United States, real appetite. I mean, there's been talk. I mean, we're already at 2020. How many of you remember Vision 2020 from the chemical industry? We passed 2020 and it didn't happen. That's where I met Ken Seddon. Another place I met Ken Seddon was 1997 in Denver, where the chemical industry was meeting to put together, hi Mara, I see you now, to put together their Vision 2020 statement about how the chemical industry was going to be green and sustainable by 2020. No.
Okay, thank you very much. Any other question? Professor Mara Freire, great pleasure to have you here.
Thank you so much. Thank you, Professor Rogers, for the amazing talk, as always, and very inspiring talk. And regarding and taking into account that our story and how we are doing it, we are also having some advice, and you have been an inspiration, and but we are following a different strategy, because our main goal now is not to find capital ventures, because then they will have the decisions, they will make the decisions. We are trying and we are fighting for funding and government funding and at European Union, because like that we can have some control on the decisions, and because there is a long path away. But your strategy is slightly different, right? Because you already have capital ventures inside.
No, I think that's kind of the point that I was making. When Mary Signum was formed, and we owned a very small portion of it, no decision making. They took it. I was happy, transfer it and let technology go. That type of funding is necessary to do it. But since they failed, in my mind, well, no, they failed. We're going back to the same model that I've talked to you about. So last week, I'm very optimistic about 2021, by the way, really, really optimistic. And last week, we got a $500,000 grant from the Department of Energy to develop our chitin nanofibers mask. And that's the only way that companies like yours and mine can go at this stage. I think one of the differences that I hope that you're doing is to focus on a high value product. I've had to not focus because I've had to look at both the supply, the extraction, and then what could be 10 million products, and which one are you going to choose? So I'm choosing to try to build that platform and then to be able to spin it out. So got a government grant for 500,000 over 2 years for one product. If I can get 5 or 6 more of those for 5 or 6 more of the products, that will also help fund the work to do the extraction. And for the extraction, I'm going to the state of Alabama and the government and saying this would be economic development for Alabama, which is a poor state. And so you have to think about all the different areas to go, but you really have to stick with it.
Yes, yes. Thank you.
Thank you very
I
much.
think my first IMO meeting was in Porto. I think my first was the first IMO, IMI, was it in Porto? It seems like I remember flying from a meeting in Australia to Portugal. It's a long time ago.
Professor Steven, I am not able to raise my hand, but can I?
Yeah, just go ahead, Professor Sonia.
Okay, thank you very much. First of all, thank you very much for the webinar organization, Steven. And of course, Professor Robin Rogers, thank you very much for the inspiring presentation as always. And this is quite recurrent in the comments from the audience because it's true, actually. I have one question, which is like more a direct question, which is devoted to the different products that we are trying to develop with the chitin. We saw in your presentation different products and with different focus or focusing different sectors of activity. So in this sense, and this is just to collect your opinion, what do you think from your experience and from all the talks that you have already so far with the different people from different fields? Which is the sector of commercial activity in which you think that the chitin will convince people first? Thank you.
So I'm not going to be, I'm going to try not to be too flippant here, Sonia, but the reality that you and I both face is that the product that brings in the money is going to be the one that the grant is going to be the one that gets the focus. So as of 2019, I started thinking, where would somebody want a textile or a fiber that was antimicrobial, antiviral, could be loaded up with stuff? And I really think if we were geared up to do it, we would be developing face mask material right now. And we have my daughter's company, Ionic, got a grant from Wyoming to do that because there's a need. And that's the simplicity of the approach that I have. There's a need, you have developed a platform technology, go out and get the money to meet that need. So for chitin, I would say Army uniforms, I would say surgical gowns, hospitals, filtration materials. The grant that we got from the Department of Energy was actually for extraction of lithium. So how can your materials be used? Now, if you, on the other hand, want to do it like a company would do it instead of academics like us, then you would find one market with a market pull, and it might be medical sutures, anything to do with medicine, and bandages, those types of things. And then you would just focus all your time just on bandages or sutures, and then you would be getting support from medical funders, and ultimately you'd probably get larger investments because the return is higher. I wouldn't focus on a food additive. I tell you one that I would be a nutraceutical. So nutraceuticals have almost no regulation, and they make a fortune. But I'm not, I am interested in money, and any business has to be, but what we're doing with chitin and the extraction, it really does feel like it's more about sustainability and economic development for, new ways. We're all going to have to find new ways to do things in the future.
Okay, thank you very much.
Thank you very much. More questions down in your, here we have one question. What about the FDA approval of the surgical bandages? It took a lot of years to previous companies to get it.
Yes, so for certain of the medical applications, you are going to have a lot of investment to go after a lot of that money. You will have to go through trials. You will have to get FDA type approvals for these types of things. But I want to remind y'all about one thing about chitin. You know what the world is like today. You probably follow politics in the US. I wish I didn't. But there is not a single chitin supplier in the US. So what if the government said you have to get all of your raw materials from the US? There's only one way I see to do it, and we're ready to do it. And while you're also looking at the bandages fibers, that's where you take it to a certain point, and if you can show you can get into that market, then you get a lot of investment. Think about a pharmaceutical. There was an ionic liquid pharmaceutical that got, I don't know, $70 million of investment to go into phase 3 trials. This was several years ago. I've talked to y'all about it many times. But the point is, if you have something that will work, that will get into those markets, then you can get the investment to proceed with it. And as far as I'm concerned, if somebody wants to, raise $70 million and take one of my things and give me a little money and send me home, I'm fine. I'll just invent something else.
Thank you very much. I think we have a space for one more question. I think I remind you that down in your Zoom panel, there is a button, says reactions. Then you can raise your hand. Professor Joao Coutinho, I think.
I didn't see
Yes,
George
if
ask a
it's
question.
about the last question, I would like to throw a challenge to Robin. Robin, I feel that the Ionic Liquid community is getting a bit down, and I'm not just talking about the inability of many of the processes that we developed in the lab to find their way to commercialization, but even at the university, at the academic level, just at the research level, I don't think things are as boiling as they were. Don't you want to leave a message, an encouraging message to all the young people around here? And there are quite a number of them in this meeting today, about the future of ionic liquids.
The encouraging message for me is to go back to the beginning, what I consider to be the beginning of the field. There was a reason that ionic liquids really got a lot of attention. And that reason was this one, green chemistry. And I think that when you get down in any academic discipline, it's because you can't tie the work that you're doing to something important. Maybe something important is really pushing the boundaries of science, and that's exciting. Maybe something is important, though, is also making a difference to society. I thought my message was encouraging, actually. If you want to make a difference, you can. Ionic liquids can do many things. They are many things that other solvents or materials are not. But you have to be willing to take the risk to go explore those. I don't want to make anybody upset by giving an example, but you don't want to be the 100th person to write a paper on a certain topic that happens to involve an ionic liquid. And that's where people can get turned off and a little boring. I'm encouraging young people to think broader, to think not only what it is that's interesting about ionic liquids, but how can you use them? And my motivation from the beginning is, how can I use these in society? And believe me, over the 20 years, 30 years that, wow, how many is it? It's a lot. Over 20 years working with ionic liquids, society in the United States is actually starting to catch up with the idea of sustainability. As they catch up, they're going to want things they didn't know that they wanted. The ionic liquids community worldwide is ready, should be ready, to provide new science and new technologies when the time is right. And I really think the time is here. Don't be discouraged. I love what I'm doing, and I think it's important, and it's just the way that I do things. But goodness, the ionic liquids field, though, Joao, it fights too much with itself, and maybe some of the things are boring now. We need to always look for what's exciting.
Thank you very much. I think one more question. Yeah, the last question.
I have one question.
Okay, Elsie.
George.
Yeah, I saw Jorge and Elsie. Jorge and Elsie.
Yes. I have a question. I heard you saying that you were working on a nutraceutical product based on chitin, because I've seen chitosan products on the pharmaceutical industry, but I've never seen chitin-based nutraceuticals.
Well, chitosan is chitin that's been deacetylated. So it starts with chitin, and then they deacetylate it, and then you can break it down into glucosamine. Now, if you look up the market for glucosamine, which is the repeating unit of chitin or chitosan, you'll find that it's huge. What you would have to do is to be able to develop a process to go from the polymer to the monomer, and that might be a little expensive. Maybe you could use enzymes, but you'd have to develop that process. On the other hand, and I say this lightly, please don't do it. With nutraceuticals, you could just make a claim that it burns fat, dissolve some up in solution, and sell it. As long as your claims are not, you will lose fat, but you just call it a fat burner, apparently you can sell it. I would hope that, well, that's a good point, Elsie, that I didn't make. If I wanted to only make money, that's easy. But if I wanted to make a difference, that's not so easy.
Okay. Thank you very much, Professor Jorge.
Thank you, Dr. Rogers, for the amazing presentation.
Where are
It's
you
always
today?
good to hear about ionic liquids. And I have a question. And also, I'm very happy to see that soon it will have a space in Tuscaloosa to create small companies. I have a small question that is related that you starting probably around 20 years ago working with cellulose, and I'm asking to you that do you still believe that ionic liquids can provide solutions for the cellulose related products? Because now you are talking about other type of biopolymers like chitin, chitosan, but we know that linear cellulosic materials are still very important to produce polymers and biopolymers. And I will ask you that is if you do you still believe that ionic liquids will provide solutions for cellulose-based materials?
I absolutely do, and thank you for asking the question. It's a strategy. So the reason that plastics got where they are, and I didn't emphasize it enough, is that at the beginning they were high-end, expensive products, jewelry, kitchenware, and it was only after there was a market and you got to the supply issues. So if we're going to work with an expensive process, which it is right now, of ionic liquid dissolution and recovery of the ionic liquid, paying for makeup of the ionic liquid as you lose it through the process, then the product has to be higher value. So I'm focusing on chitin because it's higher value. If you want to buy cellulose, you can buy it from chemical companies now, and most of the things that you would use it don't need the high molecular weight. You can, that's known. Doing this with chitin is not known. It's high value. There's no supplier in North America, I don't believe, not just the US. So the strategy was, once you've worked it out, my goodness, if I had 4 or 5 chitin extraction plants running, the cost of the ionic liquid would be dropping. I'd probably be making my own. I'd have worked out all the recycling issues. I'd have worked all the heat management issues. Everything would be worked out. And then to take that fully matured process and start doing cellulose would be a no-brainer. And by the way, they're doing some great work on cellulose textiles in I think it's Herbsixta in Finland right now. So there's no reason that process can't work and bring out a lot of new products. The key is, can we get the cellulose and the chitin cheap enough so that they really can replace plastics?
Yeah, thank you very much, Professor Robin. I think that I will read the last question, right? How is the production cost of chitin compared with ionic liquid, produced with ionic liquid compared to conventional methods?
I get that question a lot, and it's really a false question. It's not a bad question; it's a good question, but it gives a false impression. So the cost of the process is not comparable. It's incredibly expensive at the moment because nobody's done it to extract the shrimp shell using the ionic liquid. The chemical process is dirt cheap. However, you don't get the same product. I get a different product. You cannot get the chitin of the molecular weight that I get by using the pulping process. So you have to differentiate the product. If you just need chitin, then go buy the cheap stuff. As long as you're willing to put up with the guilt of using a process which is environmentally damaging. So the pulping process with all the chemical waste and everything else. They tried to put a new chitin plant in Louisiana maybe 5 years ago or so, and they couldn't get a permit from the EPA. This was before Donald Trump. They probably could have the last 4 years, but they were unable to get a permit because it's dirty. So yes, however, the cost of making plastics was incredible compared to growing a tree back in the 1920s. But today, plastics is dirt cheap. So you cannot look at the cost today. You have to be able to project what will the cost in the future be.
Okay, thank you very much. I would like to say thank you very much, Professor Robin Rogers, for being here with us, for sharing your thoughts with us in this webinar. We had today more than 130 people. I imagine a room full, plenty of people, right? Clapping now at your presentation. we cannot do that now, but you can imagine that. But this opportunity is nice because we can meet a lot of people, you know, more than we can do it in a congress. I also would like to say thank you very much all the audience for being here today. I would like to say that also we will continue with these webinars. The next webinar will be the 18th of February with Professor Jorge Brandao Pereira, who is here with us today. You are all welcome and you will receive the invitation as always. So, well, nothing else to say, and thank you very much, all of you, and especially Professor Robin Rogers.
Thank you. Bye, everyone.
Thank you very much. Take care, everybody. Stay safe.
Bye. Thank you.
Yeah, stay safe.
Thank you, guys.
Bye.
Bye bye.
See you.